Good afternoon, everyone. I am Rishabh, and today we'll be discussing about the discussion week 8 of the course Introduction to Cell Biology. And in this lecture, we'll be discussing about the nucleus and different components of the nucleus and the cell division and how it takes place. Okay, so let's start. So, giving a brief introduction about the nucleus, it is basically a well defined structure that is present mostly in the eukaryotic cells. Okay, as we remember, the basic differentiation between the prokaryotic and the eukaryotic cells is the presence of the nucleus. Okay, because in the prokaryotic cells there is no nucleus present. Okay, and they also have a particular they do not have a particular well defined region to actually store the DNA. Okay, so in eukaryotes there is a well defined region that is called as nucleus. They store the DNA, that is major storage of the genetic information. Okay, and in case of eukaryotes we have certain exceptions. For example, RBCs, they initially have the nucleus, but during the course of their development, they leave the nucleus because they have to carry oxygen. So, to make space for the oxygen carrying capacity of hemoglobin, the RBCs leave their nucleus. Okay, so that is why they are called as enucleated cells. And then this is the largest and the stiffest organelle in a eukaryotic cell, and it is actually identifiable by a light microscope. Okay, then the average nucleus diameter is around 6 micrometer, and it occupies around 10% of the total cell volume. So this is that much large organelle that is actually occupying only the 6% of the 10% of the total cell volume. Okay, and so this is a nucleus structure basically, and this central region is the nucleolus. This covering of the nucleus is the nuclear membrane or the nuclear envelope. Then we have nuclear pores that actually allows the movement of the protein to and fro from the nucleus or to the cytoplasm. Okay, then what happens? So this is the nucleus covering, the nuclear lamina, that is the nuclear membrane, and then we have nucleoplasm. So this is the nucleoplasm is basically the solvent or the substrate basically in which the different nuclear components are arranged. So, like cytoplasm, in which all the organelles are present, the nucleoplasm is a space between in the nucleus where all the nuclear components are arranged. Okay. Then we have the euchromatin and heterochromatin region. So, this is the part of the DNA that we'll be discussing in later slides. Then we have another part of the DNA, that is nucleolus. Okay. So, nucleolus is a well defined region where the major rRNA synthesis takes place. Okay, so this is the case, and then we have this nuclear envelope. Nuclear envelope is continued by the endoplasmic reticulum that contains ribosomes, and that is called as a rough endoplasmic reticulum. Okay, so this is the basic introduction about the nucleus. If you guys have any doubt, you can ask. Okay. Okay, so let's move on to the next question.
Sir,
Yes,
hello.
yes.
Can you please elaborate the uh, mm, nuclear pore?
Okay, so actually we'll be discussing that in a couple of slides more. So uh, I think that then I'll explain the structure and how it functions. So will that be fine?
Okay.
Okay, so let's move on. So the first question is, which of the following statements about euchromatin is true? So what can be the correct answer? Okay, any more answers? Okay, any more answers? Okay, any more answers? Okay, so the correct answer is option number four. That is, euchromatin is less condensed and a gene-rich region. Okay, so now we'll be seeing just 1 minute. Yes, just 1 minute. Actually, my laptop hanged. So 2 minutes. Yes. So in DNA, we have two regions. Okay, euchromatin and heterochromatin. Okay, so what happened, how they are distinguished actually, when we stain DNA with a stain called as GEMSA stain. So GEMSA stain actually stains the nucleus. It's a positively charged stain. And what is the charge on the DNA?
Negative charge.
Yes. Okay, why it's negative?
Because of phosphoric acid. PO3 2 minus.
Okay, so because of the phosphate group, this DNA has a negatively charged. Okay, so this DNA is stained with all the dyes that have a positive charge. So one of them is GEMSA stain. So when we stain this DNA with the GEMSA stain, we observe two types of regions in a particular DNA molecule. Okay, one of the region is less stained and the other region is more stained. Okay, so the less stained one had a, like, it was observed that this less stained region is comparatively less condensed, and this is actually transcriptionally active region. Okay, and the region that is more stained is actually more condensed, and this is transcriptionally inactive region. Okay, so now how it is important in a cell basically. So what happens is whenever we saw the you guys remember the genome organization. Okay, so in that case, what happens? We have the histone core that wraps around the DNA, and that is a very highly folded structure. Okay, to arrange in a small nuclear space. So like this. So this is a histone core actually. So when a gene has to be transcribed from this particular region, so what will happen? We have to actually open this histone core so that the DNA that is wrapped around this histone core would be accessible. Okay, to the proteins that are involved in the expression of the gene. For example, here we have shown the RNA polymerase. So RNA polymerase helps in transcription of the gene. Okay, so producing the RNA molecules. So to for a gene to be accessible, it needs to be opened from this histone core. Okay, so what happens is in a highly transcriptionally active cell, what will happen? There would be more amount of euchromatin. Okay, so that means all the genes that produce proteins will be present in a euchromatin region where they will be actually doing the expression of the genes. Okay, so euchromatin in simple terms will be the region where genes are present, and these genes are actually expressing proteins. And heterochromatin contains the region that does not produce any protein, and it is expression inactive. Okay, for that particular cell. So till here the explanation is clear. Okay, so now what happens is in a cell, in a nucleus, for example here on the right side, there are two regions that are present that will be shown here. This light gray region, this is actually the euchromatin region. Okay, and the black region that is actually near to the nuclear membrane is actually heterochromatin. Okay, so it was observed that in a nucleus there are two types of regions, that is euchromatin and heterochromatin, and they are also allocated some space in the nuclear membrane, some specific space rather. Okay, so for example, all the heterochromatin is actually arranged near to the nuclear membrane except for a few, and the rest of the region that is here, so it is present distributed in a nucleus. Okay, so what happens? It was given a term that is a bowl that is full of meatballs. Okay, so these meatballs actually represent the regions where the euchromatin is present, and the rest of the region where the euchromatin is not present, that is called as heterochromatin. So in a nucleus, the region for both eu and heterochromatin is divided. Okay. So, any doubts in this particular question? Okay, so let's move on to the next one. So insulin is produced primarily in pancreas. So why it is not produced in the eye? So what can be the correct answer? Okay, so some of them have second. Any more answers? Okay. Okay. So the correct answer is option 2. Okay. So remember one thing. We have how many chromosomes in humans? 22 autosomes plus XX or XY, that is in the that is the sex chromosomes. Okay. This chromosomes contain all the genes that are responsible for production of every protein in the human body, and each cell of the body have these particular chromosomes. Okay, so there is no cell in the body, human body or any animal body, that doesn't have these number of chromosomes. If that is the case, then that would be a diseased condition. Okay, so what happens actually? There are some proteins that are specific to the organs. For example, here we'll take the example of insulin itself. So insulin is responsible in lowering the blood glucose. Okay, and it is produced in pancreas. There is no other site of production of insulin. Okay, so what happens is in other organs, for example, in liver, in eye, in lungs, in stomach. I am taking some examples. Okay, in all these cases, the gene of insulin is present, but there is no protein production there. Why? In that case, what happens? The body or the cell has evolved with a mechanism that each gene specific to any organ will not be produced in the other organs, and that is due to the post transcriptional or post translational regulation. Okay, and so what will happen if any other gene that does not have any function in that particular organ will not be produced in that particular organ? Okay, because the cell will actually arrange and arrange the gene in the heterochromatin region. Okay, so heterochromatin region would be transcriptionally inactive, and it will not it will help the gene to not express in that particular organ. Okay, is the explanation clear? Any doubts? Okay. So shall we move on to the next one? Okay. So what is the role of nucleolus in a cell? So what can be the current? Okay, post transcriptional. Okay. Just 1 minute. So what happens is you know, from DNA to RNA and from RNA to proteins. This is how the expression of a gene happens. Okay, so from DNA to RNA we have a process called as transcription, and we from RNA to proteins it is a process called as uh, translation. So while the DNA produces the RNA, there is a level of regulation that happens here. Okay, so what happens is, if the gene is actually if the cell doesn't need that particular protein, so what will happen, it will actually regulate this process so that it will not be able to produce that particular protein. Okay, or it decreases the production of that particular protein. Similarly, if the cell doesn't need a particular protein but it needs the RNA of that particular protein, then the regulation is also present in that this step also. Okay, so this is how a cell maintains these particular regulations so that they can actually regulate the expression levels in that particular organ. And this is also the case with the heterochromatin region gene present. Okay, so this is the case, if I am able to explain. Okay, so the next question, what is the role of nucleolus in the cell? So what can be the correct answer? Three. Okay. Yes, the correct answer is option 3, that is rRNA synthesis. So what happens is, in a cell, this is the nucleus, and there is another region in the nucleus that is highly condensed, and this region leads to the synthesis of rRNA. rRNA is actually the major component of RNA that is being produced. Okay, so out of the three major components of RNA, that is mRNA, rRNA, and tRNA, rRNA is most abundant. Okay, so it is actually required in high amount in a cell. So that is why there is a specialized region in a cell, in a nucleus, that leads to the production of only the rRNA. Okay, so any doubts in this particular question? Okay, so let's move on to the next concept, that is the nuclear pore complex. Okay, so just one. Hmm. So, what happens is, in case of the nucleus, there would be some mechanism by which the proteins that are present in the nucleus and will actually come to the nucleus during development or during any process. So as you can see here, the DNA is present in the nucleus, the RNA is produced in the nucleus, and then it is transported out where it takes where the protein synthesis takes place. Okay, all these functions, for example, takes place with the help of proteins, and these proteins are produced in the cytoplasm. So they need to get entry into the nucleus. So, and the nucleus, because it's a storage component, storage organelle. So what happens? It has a regulated movement of any of the components either from the nucleus to the cytoplasm or vice versa. Okay, so this movement of the any molecule from the nucleus and to the nucleus happens with the set of promoters, set of transporters that are a part of nuclear pore complex. Okay, so this nuclear pore complex actually have certain proteins that act as transporters that allow only entry of certain molecules or exit of certain molecules that have certain signals. Okay, so the proteins that enter into the nucleus have a nuclear localization signal that is called as NLS. So any of the proteins that is entering into the nucleus have this particular nuclear signal. Okay, that is recognized by the proteins, and those proteins actually enter to the nuclear pore complex and they enter to the nucleus. Okay, so as you can see here, the basic structure, I'm not going into that detail, but the basic electron micrograph structure of a nuclear core complex is like this. Okay, so this is the nuclear region and this is the cytoplasmic region. So, first I'll talk about the pore part, that is actually a hole that is made in the nuclear membrane. So, this pore is actually surrounded by an outer ring, okay, that creates a pore in the nuclear membrane. Then we have a part of the same ring is the transmembrane ring that is present in the membrane region. So, as we have discussed, the membrane is actually hydrophobic. Okay, sorry, amphipathic. So the main mid part of the membrane would be hydrophobic. So this transmembrane region would be high in the hydrophobic amino acid residues that actually maintains the free energy change. Okay, then we have another set of proteins that make the inner ring of this particular pore complex. Okay, so there are three regions of this pore complex. The one is the outer ring that is actually in contact with the cytoplasm, the transmembrane ring that is maintained in the membrane of the nucleus, and the inner ring that is present in the inner ring that is present in the that covers the inner leaflet of the nuclear membrane, and it is in contact with the nucleus. Okay, in the nucleoplasm basically. Then we have another components, that is first is the cytoplasmic filaments, so that actually help in catching the proteins or the importers that help in importing proteins to the nucleus. Okay, and then we have a nuclear basket. So nuclear basket also plays a similar role. The proteins that or the RNA that needs to be that needs to be secreted out of the nucleus needs to be first come in contact with this nuclear basket, and then it will actually lead the way out. Okay, and all the proteins that come in and out of the nucleus are actually transported through a central transporter. Okay, so this central transporter is actually a protein complex that is covered by this ring complex, so that all the proteins can interact and go in and out of the nucleus. Okay, so any doubts in this particular structure? Okay, yes. I'm going to ask that question only. So, what happens is, like the smallest protein that actually come from the nucleus to the cytoplasm is I don't remember, so I have to check on that. Okay, so this is your question actually. So what is the smallest protein that go from the nucleus? Okay, the largest protein or the largest cargo that come from the nucleus to the cytoplasm is the large subunit of the ribosome. Okay, that is the largest cargo that is driven out of the nucleus from the nuclear pore. Okay, so because the large subunit of the ribosome have the ribonucleic rRNA plus the proteins. Okay, so what is actually the large subunit? That is the 50s. Sorry, 60s in case of humans. Okay, so this 60s is the largest cargo that actually is driven out of the particular nuclear core complex. Okay, any doubts? So, as per the literature, around the diameter, the largest diameter of compounds that can actually transport through the nuclear core complex is around 40 nanometer. Okay, so that includes the larger subunit of the ribosome. Okay, so this is the case, and in the on the right side, this is shown different protein complexes of the nuclear core complex, and in the different different organisms. Okay, so what are the different proteins that are present in the different regions of the nuclear core complex? Okay, so there are numerous proteins because it is a highly complex structure, so I'm not going into that detail, but these are the different protein complexes that are present in the nuclear core complex. Okay, so this is the case with the nuclear core complex. Okay, shall we move forward? Any doubts till now? Okay, so the next question is which molecules require active transport through the nuclear core complex? So, what can be the correct answer? Okay, any more answers? So the correct answer is option 3, that is large proteins and the ribonucleoprotein complexes. Okay. So this is a nuclear pore complex, and as I said, that the proteins that have a size of less than 40 nanometer actually can pass through the um, nuclear pore complex. But there are two types of transport that takes place through the nuclear pore complex. First is free diffusion, and the second one is active transport. So there is a cutoff. If the molecules are less than 50 kilodalton in size, so they can actually freely diffuse through the nuclear pore complex. They do not require any energy or any subsequent proteins to get involved and to uh, able to transport through the nuclear pore. So, for example, metabolites, ATP, and small proteins that lie under the less than 50 kilodalton size parameters, they actually transport through free diffusion. And the proteins that are greater than 50 kilodalton size, that is large protein complexes, the the ribonucleoprotein complexes, RNAs. So these actually include the size of greater than 50 kilodalton, and they require the active transport. Okay. So, and what is actually meant by the active transport? They would need some proteins that will bind to them, and they will actually help them to move out of this nuclear pore complex. And the one of the protein is actually ran GTP. So it is actually a G protein, so it is bound with GTP. Once it binds to the protein that needs to be transported through the nuclear pore complex, this GTP is converted to GDP. So here, because the GTP conversion to GDP takes place, it is an energy consuming process. So once it's an energy consuming process, this leads to the active transport. Okay, so this RAN GTP is an important molecule that helps the large protein complexes to move out of the nucleus and to move in of the nucleus. Okay, so any doubts in this particular question? Okay, if not, then we'll move on to the next one. What is the function of importance in the nuclear transport? So, what can be the correct answer? Okay, any more answers? Okay, we'll explain it after the question. Mhm. Any more answers? So, what happens is, if, for example, this is the nucleus and this is the cytoplasm. Okay, so if we have to move a particular protein into the nucleus, that process will be called as import. Okay, and if we have to move anything from the nucleus to the cytoplasm, it will be called as export. Okay, so this is exporting of a protein from the nucleus and importing of a protein to the nucleus. Okay, so the proteins that help in this import process is called as importance into the nucleus, and the proteins that help in the export process from the nucleus, these are called as exportins. Okay, so the importance actually help to import proteins into the nucleus. So the correct answer is option 2. Okay, and here is a generalized mechanism how importance and exportins work. So I have just now told you about the protein called as RAN-GTP that helps in the active transport of the protein. Okay, so what happens is this importin is carrying a import cargo. Okay, now this important molecule is actually now if it has to import it. So what will happen, it actually binds to the nuclear pore complex, and now when it enters it, and the ran GTP protein will now bind to the particular cargo and release the importance into the nucleus. Okay, same thing happens when a exportin works. So exportin binds to the export cargo, then it uh, leads to the release of the cargo once the RAN GTP hydrolysis takes place. Okay, so the GTP is converted to GDP and the hydrolysis takes place. This leads to the release of the export cargo in the cytoplasm. Okay, this is how both importance and exportins work. So, any doubts in this particular question? Okay, so let's move on to the next one. The complex of proteins associated with the inner and outer nuclear membrane is called. So, what can be the correct answer? Okay, any more answers? Okay, any more answers? Okay. So the correct answer is option 3, that is the link complex. So here what happens is this is the link complex. Okay, so this connects the this is the inner nuclear membrane, this is the outer nuclear membrane, and it connects both the inner and outer nuclear membrane with the nucleoskeleton and the cytoskeleton to get a fixed shape basically. Okay, so the full form of link is linker of nucleoskeleton and the cytoskeleton. Okay, so it has certain proteins. For example, these are the intermediate filaments, and this is an actin filament in the cytoplasm. Through the intermediate filaments, there is neosprin 3 that connects it to plectin. Okay, so this is one of the proteins that help in binding to the intermediate filament. Then neosprin 2, 1 and 2 actually help the binding of these proteins to the actin filament. So this was from the cytoplasmic side. Now there are two proteins called as SUN1 and SUN2 These two proteins are bound from the outer membrane of the nucleus to the inner membrane, and this extension is bound to lamins. Okay, so this lamins are a part of the nuclear skeleton, and this nuclear skeleton, so this is actually an intermediate filament. Okay, so this is a part of the nuclear skeleton, and the sun 1 and sun 2 proteins bound to lamins bind to this neosporin 3 and neosporin 1 and 2 so that they can attach to the cytoskeleton. Okay, so this is how they both arrange in a connection way from the cytoskeleton to the nucleoskeleton. Okay, so any doubts in this particular question? Lamina is basically a, okay, so in this case it's actually a complex of the lamins. Okay, so lamins make a thick structure inside the nucleus that is called as lamina. Which remaining complexes? Oh, sorry. In this case, yes, yes, sure. Okay. So the snare and the clathrin complex is actually related to the vesicles that are imported during endocytosis. Okay. So this snare complex helps in movement of the vesicles from Golgi, and clathrin complex actually helps in endocytosis. Okay, so clathrin mediated endocytosis is one of the most important part of the endomembrane system. Okay, and the COG complex is actually center of Golgi, so it helps in the arrangement of the Golgi complex. Okay. Any more doubts in this particular question? Okay, so let's move on to the next question. Outer nuclear membrane is continuous with. So what can be the correct answer? Okay. Yes. So the correct answer is option 2. That means the this is the outer nuclear membrane, and this is continuous with the endoplasmic reticulum. Okay. So this is the case. And the rest of the membranes are separate from the nucleus. So let's move on to the next question. Lamins are, what can be the correct answer? Okay. Any more answers? Yes. So the correct answer is option 1. That is, these are the cytoskeleton proteins present inside the nucleus. Okay. So this is a arrangement of the lamins. So these are basically distributed in the nucleoplasm and helps in the arrangement of the chromatin molecule, the different proteins that help in. But there is a bunch of lamins that is present just inside to the inner nuclear membrane. Okay, this bunch of lamins actually supports the nuclear membrane. Okay, so this is the major role of the lamins. There are two major type of lamins that are present in the nuclear membrane. In the nucleus. That is A type lamins. Okay, so in that case, there are two lamins, lamins A and C, and these are in present in the differentiated cells. Okay, and B type lamins B1 and B2, they are found in all nucleated cells. Okay, so why this major differentiation between the two types of lamins? Because in differentiated cells there is no cell division. Okay, that means their nucleus would be more rigid in nature. Okay, so these lamins, that is type A lamins, they actually help in making the nuclear membrane more rigid. Okay, while this lamins B1 and B2, these are present primarily in all the cells. So that is not a like division. Okay, so any doubts in this particular question? Okay, so there are certain diseases that are also associated with the mutations that can be created in lamins. Okay, so let's discuss that in one of the questions. So, LMNA gene that codes for why they are not present in the human genes. They are present in the human genes also. Lamin A and C are present in human genes. Sorry, human cells. No, differentiated cells means where there is limited or no cell divisions. Okay, for example, what we call it in humans, it is terminally differentiated cells. Okay, so for examples, the best example is neuron. Okay, does it divide? Yes. So, what happens in case of neurons? There is no division. Okay, so differentiated cell doesn't mean that it will not be present in humans. Okay, in humans also, not every cell divide after maturation. Some of the cells divide for their regular tear and removal of the cells, but there are certain cells, for example neurons, they do not divide. And if there is any damage to this particular neuron, that cannot be repaired also. If there is something damage that will not be repairable, then that will cause a disease. Okay, is it clear the doubt? Hmm. Okay. So there is the lamin A that we were talking about is encoded by a gene called LMNA gene. Okay, so the mutation in this gene causes which disease? So we have got one answer already. The other ones, Can you guess? Okay. So yes, the correct answer is option one. And in this case, this is actually a premature aging disease. Okay. So this is how the appearance of this particular disease looks like. C disease. Okay, I'll explain it. So like in this case what happens, there is the increase in the size of the skull, and the patient suffering from this is actually looks like a grown up individual in a very short age. Okay, as you can see in the movie Pa, the Amitabh Bachchan also had this particular disease. Okay. So what happens is, if this particular lamin A gene is mutated, so it will actually lead to the mechanical defects. That means it will not be able to hold the nuclear membrane properly. Then it will also alter the chromatin organization and genomic stability, because the chromatin actually is attached to this particular lamins, the heterochromatin basically. Okay, that is why it is part of the transcriptionally inactive region. So, if lamin gene is mutated, that means the heterochromatin region that is involved in the stability of the nucleus will be hampered. Okay, this will lead to the what will happen? The lamin A gene is mutated. It leads to production of a mutated lamin A that forms a aggregated structure called as progerin. Okay, so there is aggregation in this particular case. For example, here as you can see, this is progerin. So this progerin inhibits or alters various signaling pathways also. Okay, and it will also like alter the regulation of the stem cells. So this is how different, like from mutation of a single gene, there would be multiple defects that will be seen in case of the mutation from lamin A gene. Okay, so any doubts in this particular question? Yes. Okay. So in the pro-metaphase stage, the nuclear membrane is completely broken down. Then the chromosomes are continuing to condense and they are arranging themselves in the central plate. Okay. Then we have this the spindle fiber actually is formed from the centrosome, and they are attached to the kinetochores so that they can attach to the chromatid molecules. And then the centrosomes, that are the production of the microtubules, they reach the opposite poles of the cell for the cell division. So these are the major attractions of the prometaphase phase. Then from the prometaphase phase we move to metaphase. Okay, so as the name suggests, meta, this chromosomes are arranged at a central plate in a in the cell, where they are attached to the kinetochores through the centromere. Okay, so centromere is actually a point where the two chromatids are attached in a chromosome. So the center circle is called as a centromere. From this centromere, the kinetochores are attached. Okay, and so they are lined up at the central plate, and each sister chromatid is attached to a spindle fiber. So these spindle fibers are attached to the kinetochores in the centromere of the chromosomes. Okay, so the major attractions of metaphase is they are arranged in a central plate. The mitotic spindle is attached to the kinetochore, attaching to the centromere of the chromosomes. Okay, so now from here the major division starts. This was the arrangement. Now from here the division starts. So next comes the phase called as anaphase. Okay, in anaphase what happens? The mitotic spindle now try to move to the opposite poles. So what will happen? The centromere actually divides from the chromosomes. Okay, so the cohesin proteins, that is the part of the centromere, they divide or they break down, so that the sister chromatids or these chromatids are separated into two chromosomes. Okay, so they divide, then they are called as chromosomes. They start pulling towards the opposite pole because the spindle fibers that were originated from the centrosome are start actually pulling towards the end of the cell. Okay, then these non-kinetochore spindle fibers lengthen and elongates the cell. Okay, so they actually elongates this particular cell so that it will actually help in division of this particular cell. Okay, then the last phase is the telophase. Okay, so in telophase what happens? These chromosomes, the separated chromatids, reach the opposite poles and they start decondensing. Okay, then the nuclear envelope and the nucleolus reappear. Lastly, the mitotic spindles also break down and they reach the centrosomes. Okay, so this is basically the recycling phase where all the things which have disappeared now reappear into two in duplicates so that the two cells can be formed. Till now, we have seen the difference or the phases of the division of nucleus. Okay, so this division of nucleus, this is called as karyokinesis. Okay, now the last phase, that is the division of the cytoplasm, is called as the cytokinesis. This happens so that the cell is actually completely divided into two daughter cells. Okay, so there is a furrow formation that happens in case of the animal cells. So this furrow move from the outer to the inner but inner part of the cell, and it leads to the breakage of this particular cell membrane. In case of the plant cell, the formation of a cell plate happens, that happens from the center of the cell to the periphery. Okay, so this is the difference between the plant cell and the animal cell. In the animal cell, the furrow actually grows inwards, and in case of plant cell, the cell plate grows outwards. Okay, so this was the part for the cytokinesis, where the cytoplasm division takes place, right? So these are the different phases that have been actually captured from live cells, okay, or the cells that have been used in vitro. So these green colored regions are labeled with the microtubules. So the spindle fibers are actually being represented via the green colors. Okay, this blue region is the nucleus or the DNA component. And this red colored region that can be visible here, these are the different proteins that are associated with the DNA. So here the DNA is actually chromatin and it is starting to condense. Okay, and as it condenses, it will reach the central plate, as in case of the metaphase. Okay, so this leads to this. This is actually condensing, and this leads to this. So this is arranged in a metaphasic plate. All the DNA is arranged in the middle of the cell, that is the middle plate, and now it is attached to the kinetochore, so that once it divides, it leads to the division of the chromosome also. And then this spindle fiber actually takes the sister chromatids to the opposite poles in the anaphase. Now, after this, in the telophase, the nuclear decondensation occurs, the spindle fiber also is removed, and there is initiation of the formation of the nuclear membrane and the nucleolus. And in the last phase, that is cytokinesis, the cytoplasm division happens. So these are the basic phases of the mitosis. So any doubts in this particular slide? So basically there are different proteins that are involved. Okay, I have not gone into that detail, but there are proteins that help the different points or the different condensation, decondensation, the movement of the proteins from the movement of the chromosomes to the different plates or different parts of the cell. So these are regulated by the proteins. Okay, and these proteins actually take place in the cell cycle phase. Okay, once a cell enters into mitosis, it has to complete the mitosis. There is no doubt. But the other interface phases, we have certain checkpoints. So if a cell is actually suitable for division, then they pass the checkpoints, and there are different cyclin proteins that are involved in this particular cyclin and CDKs. So cyclin dependent kinases that are actually involved in the checkpoint phase. So these proteins are actually involved in the checkpoint phase of this particular cell cycle. Yes, it is absolutely necessary because the division needs certain amount of proteins that can play important role only in the division. Okay, not in any other process. So for that, yeah. So for that, what happens? The cell needs to prepare for the particular division, because now this particular one cell is need to be divided into two. So that means all the components need to be duplicated basically. So for that, this interface is very, very necessary. Okay, is the explanation fine? Any more doubts? So mitosis actually makes clone copy. So because all the information is duplicated basically, so they make a clone copy. Okay, so let's move on to the next question. Okay, so wait a minute. Okay, so there are major three checkpoints that are involved in the cell cycle. First checkpoint is the G1 checkpoint, that actually helps or the realizes that the cell is actually ready to divide, and it now enters into the synthesis phase. Okay, so here, like, I'm not going into that detail that which cyclin and which CDK is involved in which checkpoint. I'm now just telling you about the different checkpoints and what is their major role. Okay, so one is the G1 checkpoint, that allows the cell to move to the DNA synthesis phase. Okay, if the cell is not ready here yet, now from here the cell will again move to G1 and will grow. Okay, now from this, the second phase is the second checkpoint that comes is the G2 checkpoint. So this actually tells that uh, the cell is ready for mitosis or is ready for division. Okay, and the last one is the M checkpoint, that is if if the division is actually taking place properly or not. Okay, so these are the three main checkpoints that are involved in a cell cycle. Okay, is it fine? Okay, so let's move on to the different questions related to the cell cycle. So first is, during which phase of the cell cycle does the nuclear envelope break? Any more answers? So yes, the correct answer is option 4, that is M phase. And in Specifically in the M phase, there is prophase, where the nuclear envelope breaks down. Okay, so any doubts in this question? Yes. So let's move on to the next one. What key event occurs during the prophase of mitosis? So what can be the correct answer? Hmm. Okay, so the correct answer is option 3. So if we see different options, so chromosomes are replicated in the S phase of the cell cycle. The nuclear envelope reforms, it is in the telophase, and cytokinesis is completed. This is the end of the M phase. Okay, so the major thing that happens in prophase is chromosomes condense and the mitotic spindle begins to form. Okay, so let's move on to the next one. Identify the stage of cell cycle shown in the image given below. So what can be the correct answer? Okay, any more answers? Yes. So the correct answer is anaphase. And how it is anaphase? Because the sister chromatids has breached the opposite poles and the spindle fibers also start to disintegrate. Okay. So this is the anaphase. Okay. So let's move on to the next question. The role of dash is to aid attachment of chromosomes and the microtubule spindles. So what can be the correct answer? Hmm. Okay. Okay. Any more answers? Hmm. Any more answers? Okay. So the correct answer is option 4, that is kinetochore. So this is actually a structure how a kinetochore actually attaches to the centromere and the chromosome. Okay. So this is kinetochore microtubule. Okay. And this is the centromere through which the kinetochores are attached. And these are the sister chromatids. Okay. So here also this is the kinetochore and these are the spindle. So these spindle are attached to this kinetochore and this kinetochore is present in the centromere region of the chromosomes. Okay, and now when they are attached they try to separate it. Okay, so this is the case. Any doubts in this particular question? Histones histones help in condensation of chromatin because the DNA is bound on the histones. Lamins are a part of the nucleoskeleton. Centrioles are part of centrosome. So they help in formation of the spindle fibers. Okay. So any doubts in this particular question? Okay. So let's move on to the next one. During which phase of the cell division are the chromosomes attached to the spindle fibers? Okay, any more answers? Okay, any more answers? Yes, the correct answer is option number two, metaphase. So, in case of the metaphase, what happens? The chromosomes are arranged in central plate that are attached to the kinetochore through the centromere. Okay. So, any doubts? Okay, if not, then we'll move on to the next question. Which of the following statements about cell division is true? So, what can be the correct answer? Okay. Okay. So, correct answer is option 3 and 4 both. Okay. So, DNA condenses, yeah, just 1 minute. So, DNA condenses to form chromosomes. Then, in that case, this happens in case of the prophase only. Okay. And actin has no role during the cell division. Okay, so all the role that is takes place is of the microtubules. Okay, the other two options, that is Golgi fragments, falls back to the ER. So they actually trying to explain that with during the division the Golgi fragments disassemble and they fall back to the ER, and this would be then. Okay, so this would be then mitochondria fragments, and the fragments it fragmented are distributed throughout the cells. Okay, so mitochondria do not fragment itself. Okay, so this is also false. So option 3 and 4 are correct. So basically what happens is, I am zooming in, so this is actually explaining about the different organelles that how the division takes place in case of different organelles. Okay, because not only the DNA or the this thing or the centrosome divides, but the different organelles also are duplicated. Okay, for the mitochondria and chloroplast, they are endosymbionts. Okay, so they divide as a new organism. So they also divide from the pre-existing cell itself. So one mitochondria give rise to two mitochondria. Okay, so that is done during the G1 and the S phase. Okay, then the Golgi, as we are speaking with, the Golgi is also fragmented into two Golgi fragments. Okay, so sorry. So this Golgi is actually divided into two Golgi fragments, as we can see here. This is the prometaphase. Okay. Then during this time, the Golgi reaches the opposite poles during the metaphase. All the other organelles, that is mitochondria or the endoplasmic reticulum, starts to arrange in the opposite poles because the cell would be forming in this way. Okay. Likewise, in the telophase, all the organelles that have been disassembled during the prophase will now reach to the opposite poles and start reassembling. For example, ear also disassembles and it reassembles during the telophase. Okay, so that they can actually reach to a cell that represents the interphase nucleus. Okay, so any doubts in this particular question? Okay, so let's move on to the next concept, that is the centrosome cycle. Okay, so what happens is the centrosome is a key to the production of spindle fibers. So the duplication actually taking takes place in the interface itself. Okay, no duplication of any organelle takes place from the prophase. So everything duplicates within the G1, S, and the G2 phase. Okay, so moving on. So centrosome is helps in the production of the spindle fibers. So what happens is centrosome first divides. So this is the centrosome. Okay, so this is the centrosome, and two. It has to divide into two centrioles. Okay, because it will also be divided into two daughter cells. Okay, so this centrosome give rise to two centrioles. This is the mother centriole, and this is the daughter centriole. Okay, so these centrioles actually give rise to actually nothing disappears. Okay, they are just fragmented and kept as a small pouch in a distant part of the cell. Okay, so that they do not hamper any cell division process. So all the organelles that actually disintegrate or like that disintegrate into the prophase reappears in the telophase. Hmm. Is the explanation clear? Okay, so now moving forward, so this both centrioles will give rise to the microtubules. Okay, now what happens? There is centriole. So centrioles actually, so this is the centrosome is actually a microtubule production house, a production center. Okay, so So this is production center. So actually they are in the form of a cylindrical path where they give rise to the microtubules. Okay, and here different proteins are involved. So these give rise to the daughter centrioles. Okay, I'm not going into that detail, but because it's a microtubule production center, it has that ability to actually divide because they give rise to the microtubules. Okay, so after the divide, there is a strong arrangement between the mother and the daughter centriole that is actually broken down. Okay, so that means the two centrioles are divided and they separate from each other, so that now this centrosome actually gets duplicated. Okay, in each centrosome, both these mother and the daughter centriole will be present. Okay, so now this centrosome is actually now duplicated, so there would be two mother centrioles and two daughter centrioles. Okay, so the other after the centrosome duplication, there will be a new centriole that will be formed that would also have two centrioles, that is mother and the daughter centriole. Okay, so that means in each of the case it would be considered as one. Okay, now then what will happen? It will be then the centriole engagement happens, so it reaches its full length, and now they can actually uh, form spindles, and then centrosome maturation happens. Then after the maturation, there is the centrosome separation happens, followed by the cell division. Okay, so during cell division, both the centrosomes reach the opposite poles. Okay, so this was the centrosome cycle in brief. Although there are many components that would be involved in this case, but I am not going into that detail here in this particular time period. But these is an important cycle that runs side by side through the karyokinesis. Okay, so any doubts in this particular case? Okay, so centrosome has two centrioles. How many microtubules are hence connected to each centrosome? So what can be the correct answer? Okay, any more answers? Okay, so what happens is, in case of a centriole, it is made up of okay, so it is made up of an arrangement of 9 plus 0. Okay, and this 9 is arranged in triplets. So this is for only one centriole. Okay, but in case of a centrosome, because the centrosome is needs to be answered, so for centrosome it has two centrioles. So that means 18 triplets of microtubules would be present in a centrosome. So the correct answer is option number 4. Okay. So any doubts in this particular question? Okay, so let's move on to the last part of the lecture, that is the meiosis. Okay, so mitosis we have talked about. It takes place in all cells, all somatic cells. So the chromosome number is maintained, the clone is formed, that is the cell that is formed from a pre-existing cell would be same, the amount of DNA is same, the number of organelles is same, and because all of them have been formed from a pre-existing cell. Okay, there is another division that happens in the gametes or during the formation of the gametes, that is meiosis. So in this case what happens, there is reduction in the chromosome number, because as we have seen there are chromosome pairs, that is 22 plus XY pairs. So total of 46 chromosomes are there in a human cell. Okay, so these pairs, if we talk about one of the pairs, that means 22 plus X or Y comes from, X comes from a female counterpart, while the other 22 plus Y comes from the male counterpart. Okay, that after this fusion they will form 22 plus X or Y, and this is pairs. Okay, so here from this female 23 chromosomes came, from this male 23 chromosomes came. So the total of 46 pairs are formed. The normal chromosome number in a human cell is 46. So how this 46 changes to 23 is basically answered by the meiosis. Okay, so meiosis takes place in the formation of the gametes, where the chromosome is the cell is actually divided into four cells that have half the number of chromosomes that is present in the parent cell. Okay, so this is the major advantage of the meiosis. So talking about its steps, there are two phases in a meiosis. This is meiosis 1 and this is meiosis 2. Okay, the meiosis 1 is basically replica of the mitosis. Okay, so it has prophase 1, where like in the mitosis the chromosomes condense, nuclear envelope breakdown, crossing over occurs. Okay, now here we have another term, that is crossing over. Okay, so crossing over is basically the exchange of the exchange of the genetic material between two chromatids. Okay, so between two chromatids, from the pair of chromosomes. So for example, sorry for the diagram. So this is a chromosome that came from male counterpart, and this is the chromosome that came from the female counterpart. There is a gene called A that have different alleles on both the cases. Now the crossing over happens and these alleles are replaced with each other. Okay, so now after the crossing over, what happens is this capital A comes to the male counterpart and this small a comes to the female counterpart, and this will go into different gametes. Okay, so now this is a basic uh, description about the crossing over. So this will lead to the different non-parental gametes that form during the course of the meiosis. Okay, now what happens? The homologous chromosomes are arranged in a metaphasic plate, then they divide and they form two different cells that have equal number of chromosomes. Okay, so in the telophase 1, the chromosomes have gone to the different poles, they decondense, they do not decondense. Okay, they remain in the opposite poles. The cell has divided into two cells now. They have equal number of chromosomes as the parent cell. Now these two cells now enters the phase called as meiosis 2. Okay, in meiosis 2, what happens in prophase 2? The spindle now again forms. The nuclear membrane is not formed yet. Okay, and then the similar thing happens. The new spindle formation takes place. Then moving to metaphase 2, all the chromosomes are arranged in a central plate attached to the kinetochore, and then in the anaphase 2, the centromere divides. Now after division, they move to the opposite poles, and now the chromosome decondensation happens and there is formation of two cells from this one cell. Okay, this cell that is formed contains only one set of the chromosomes. So, for example, if there are two n number that is present in the parent cell, now each cell would have a chromosome number of n. Okay, that means the chromosome number has halved in the gamete cell as compared to the parent cell. Okay, so this was a brief description about the meiosis process. So if you guys have any doubt, you can ask. So this was the last part of the lecture. Yes, interface would be same for both. Okay, just the type of cell that takes that goes into the division is different. So any more doubts? No. In prokaryote cells, the division is totally different. This is only in the eukaryotic cells that we can see. Okay, this was the last part of the lecture. So if you guys have any doubt in the presentation, you can ask. Okay, so let's close for the session for today. So thank you guys for joining in.